Now, Professor Dame Ottoline Leyser is our speaker tonight. She's the Chief Executive of UK Research and Innovation, UKRI, and Regius Professor of Botany at the University of Cambridge. The UKRI brings together the UK's research councils, Innovate UK and Research England, with a combined budget of more than £8 billion per year. Before this role with UKRI, Ottoline was director of the Sainsbury Laboratory at the University of Cambridge. It's an interdisciplinary research institute which aims to understand the systems which regulate plant growth and development. She's also worked extensively in science policy and in 2017 was appointed Dame Commander of the British Empire for services to plant science, science and society, and equality and diversity in science. She's going to bring together these research threads and policy threads together in her lecture tonight, called Plant Genetics from Mendel to Monsanto. So I'll hand over to Ottoline now.
Thank you very much indeed, and it's a great pleasure to be here, albeit only virtually. I've become sadly rather used by now to talking to my laptop, and it's not entirely straightforward because I can't see any faces to know how things are going, but I'll do my best, and I look forward very much to taking your questions at the end. So what I want to talk about is plant genetics, and in the context of the controversy over genetic modification and now genome editing in crops, and how this links through to our fundamental understanding of how plant genetics works, which is something I've been interested in for many, many years. So as everybody I think knows, GM crops are very controversial, and I think it's interesting. I've been having conversations about this for a very long time, and one of the things that always strikes me about these conversations is they all start with everybody agreeing. Everybody, I think, with very, very few exceptions, is in favour of a reliable supply of safe, affordable, nutritious food that's produced in an environmentally sustainable way, with equitably shared risks and benefits associated with that whole process. And I think it's really important to hold on to this shared goal as we think about how best to deliver it in a world where populations are rising and climate change is making the reliability of crop growth more and more difficult. So this is an increasingly difficult thing to achieve, and there are many elements that need to come together to achieve it, and one of them is contributions from plant genetics. But as I say, these have become very controversial because they've been associated with modern genetic improvement techniques such as GM and genome editing. And given that everybody starts from this common goal, and there is good evidence that genetic improvement is an important contributor to that goal, why is it that people don't like particular ones of those techniques, including GM? And I think we in the plant science community at least have started with a kind of classic public engagement deficit model for why this is. People think genetic modification is dangerous because they don't really understand what it is, and to get over that, one needs to go about explaining what it is, and then everything will be all right. And it will be particularly all right if some of the applications in discussion are things that directly benefit people, so that people can not only understand whatever risks there may or may not be, but also the benefits. And that's one of the lines of argument or discussion that have gone on since these techniques were introduced decades ago now. I don't think that's actually what's going on, and certainly in my experience of talking about GM with all kinds of audiences, that's not what I get back. What I get back is much more strongly what I would call a social identity explanation for why people don't like GM. It's perceived as epitomising profit-driven agriculture and is then associated with in people's minds, with poor food safety, with inequalities and with environmental degradation. And layered on top of that, these profit-driven approaches have become equated with a kind of science-focused reductionist idea as opposed to a kind of nature-focused holism. And you can see this just walking along the supermarket shelves, where natural is an extraordinarily important word for selling food. It doesn't actually matter how good the food is for you. If it's natural, then there's this kind of perception that it must be good for you. You can find all kinds of very strange things that are basically pure sugar, which will tell you they've got natural flavorings or something, and this is supposed to make them a good thing. Meanwhile, there is a sort of general idea that people don't want science in their food. This is a very interesting advertisement I found some time ago now, that is very explicit that scientists should not be allowed to make food because they're too kind of cold and rational and are not very interested in flavour and good things. And so this idea that natural is good, science is somehow not natural, and we certainly don't want any of that in our food, I think is part of this identity approach to people's dislike for GM. It's about big profit-driven business kind of turning food into a kind of handle-cranking factory rather than something about wholesome nutrition. And this is very much encapsulated by the kind of Prince Charles quotes, like this one: in an age when we're told that science has all the answers, what chance is there for working with the reign of nature? I believe that we need to restore the balance between heartfelt reason of instinctive wisdom and the rational insights of scientific analysis. And above all, we should show greater respect for the genius of nature's designs, rigorously tested and refined over millions of years. And this is an absolute classic example of this pitting of science somehow against nature, which I think is an interesting trend, and to me is part of a much broader concern I have about the position science holds in our society, which I think is entirely wrong. It's positioned exactly as is sort of set out in this quote, as a kind of strange activity done by, genius experts in shiny labs, and they are somehow cold people who you can't quite trust because they don't have this kind of intuition. They're not interested in nature and all of these things. It's a very odd definition that I think we need to work on, because I think it's quite inhibitory to all kinds of things in our society, let alone GM crops. So how does this relate to this kind of social identity model I described? So social identity theory was kind of introduced by Henri Tajfel and is really well evidenced psychological phenomenon where people, particularly people who feel under threat in some way, will very rapidly establish an in-group, and that in-group consists of people who agree with them. There's in-group favoritism, and part of the whole thing is the definition by the in-group of an out-group. So there are others in the out-group, and you don't like them. That's part of the definition of the in-group, and you derogate the out-group. And within the group, you all kind of agree with each other to the point that you actually embed a particular set of views. So you kind of homogenize a view within the in-group, and you polarize your that view with respect to the out-group. And this is very reassuring for people. They're surrounded by people with whom they agree, they share this common identity, and part of that is the opposite to the out-group. And this happens really quickly in all kinds of situations, and it's very well documented. And I think it's the thing that kind of underpins a lot of the concern about GM crops, where you've got people, normal people, on one side of the wall in one group, and scientists, and particularly corporate scientists, on the other side. and they are each other's in-group and out-group, and the scientists think of themselves as being terribly rational and innovative, and the people think of themselves as being wonderful, holistic, intuitive people, and the scientists would label that as irrational and Luddite, whereas the people would label this kind of rational, innovative label as reductionist and robotic and arrogant. And you can see both of those in-group and out-group identities building themselves up around this kind of controversy, and it's extremely unhelpful because it separates people. And the other thing that I think is really important about it is that it embeds issues that go well beyond what you think you're talking about, and I'll get back to that in the end. You can have this conversation based on a kind of deficit model understanding of what's going on, that all you need to do is explain what GM is and then it'll be fine, and that is so far away from what people are thinking about in this context that it has no impact whatsoever. And so we need to understand, both of us, how we're defining ourselves and the extent to which that's helping or hindering an engaged conversation about these kinds of topics. So as I say, I think that this social identity explanation, the defining features of it, are to do with a dislike for profit-driven agriculture, which is elided in some interesting way with a dislike for science-based approaches as opposed to nature. And so you can draw the same kind of barrier in the sort of science versus nature axis, where the science is considered to be reductionist and rational and in some from an out-group point of view arrogant, and from an in-group point of view kind of informed and knowledgeable, whereas the nature approach is kind of holistic and intuitive, and from an in-group point of view appropriately humble, and from an out-group point of view kind of ignorant mumbo jumbo. And it's very frustrating, because I think this is very far from what is actually going on. And I want to try and illustrate that with genetics. So genetics, is genetics reductionist, which is one of these key polarizing elements. Science is reductionist, nature, we're told, is holistic. These are opposed to one another in some kind of way. And I think this question, is genetics reductionist, is quite important, because I think the popular discourse about genetics is reductionist, but genetics is entirely a kind of holistic pursuit, and I want to try to illustrate that with considerations of Mendel, for example. So if you define reductionism as the practice of analysing or describing a complex phenomenon in terms of its simple fundamental constituents, and then you're especially keen on, it's particularly reductionist, if this is said to provide a sufficient explanation of what's going on, then that's reductionism. And genetics is quite often described in that way, particularly in the media, but also just in the vernacular. People are endlessly talking about genes for this, as though you could say having the gene for this is absolutely a sufficient explanation of what this might be, and that there is a unitary thing, a gene, that is doing this to you. Whereas holism is the theory that parts of a whole are in intimate interconnection, such that they cannot exist independently of the whole, and they can't be understood without reference to the whole. And in that sense, the whole is regarded as greater than the sum of its parts. And to me, that is a way better description of what genetics is about, because genetics, from my point of view, is not about genes. It's about the relationship between the genotype, so the sequence in your DNA, and the phenotype, what you look like. In this case, I've got some wheat plants, and they have different heights, and they have different heights owing to having different versions of particular genes, and therefore different sequences of DNA at particular places. And you could say, well, there you go. That's a straight up reductionist explanation. You're telling me that there is a change in a gene. This is resulting in this change in the phenotype. That's it. I'm saying that's the initial description of the phenomenon that you're seeing, and is just the start, because to me, the exciting thing about genetics is trying to understand how on earth that happens, how a change in your DNA sequence, which is at this nanoscale, 10 to the minus 9 meters, that's nanometers, results in a change in what the organism looks like at a meter scale. That's huge numbers of orders of magnitude, and a huge amount of really extraordinary biochemistry, cell biology, physiology in between those things. And to me, that fits much more deeply into that explanation about holism, because it's completely about how all of the parts in this system interact with one another, and how these macro scale phenomena like height emerge from the behaviors of all the in-between scales, right down to the genes at the nanoscale. Oops, no, why isn't this? There we go. And just to give you a kind of scale reference, the world is 10 to the 7 meters. So the difference between a gene and a wheat plant in scale is more than the difference between me and the world. And that's the kind of orders of magnitude of action and activity of molecules, of cells, of tissues, of organs that we're trying to understand in trying to work out what the relationship is between the DNA sequence and the whole organism. And for me, this completely started in school with Mendel, as I'm sure it did with many people. And it started with this very popular description of genetics in a which sounds very reductionist and simple, and genes for. But I want to illustrate a little bit of that multi-scale thing through an absolute classic gene that I'm sure everybody learned about in school, but they wouldn't necessarily have learned that the gene is the gene that encodes a protein that's called starch branching enzyme. And that gene is the so-called round and wrinkled gene in peas. So Mendel's first law, which I'm sure you all remember perfectly from school, which is that traits are governed by hereditary factors, which were later called genes, and he postulated that they exist in pairs in peas, as they do in humans, one inherited from each parent, one from your mother, one from your father, and these pairs, these factors, do not mix during life. So it's not that the version you get from your mother and your father talk to each other at all during life, but they segregate and are passed randomly to the next generation. So when sperm or eggs are made, the 2 copies are separated. Each sperm or egg gets 1 copy, and then when the sperm and egg fuse to make the next generation, you're back to 2 copies. And so in the context of round and wrinkled, here's a pea pod from a self-fertilized pea plant. So it was both the mother and the father, and you can see the round peas here and the wrinkled peas here. And what Mendel noticed was the fixed ratio of these traits to one another in the offspring of crosses between round and wrinkled parents. So if you take a parent that is has entirely round peas, and it's true breeding, so that means if you self-fertilize it forever and ever, you only ever get round peas, and a similar one that only has wrinkled peas, then in the first generation, the children of that cross all have got the round version copy of the gene from this parent and the wrinkled from this parent, so they have this so-called heterozygous genotype. And then if you self-fertilize this pea, then you get a quarter of the progeny will have round like this parent, a quarter will have this little r like this parent, and half will look like their parent or have the genotype of their parent. And the key additional element here is that these peas are all round. These peas are all round and these peas are wrinkled because in the Mendelian term, the round version of this gene is dominant to the wrinkled version. So if you've got the round copy and the wrinkled copy, then everything's round. So this is what you learn in school, and it's called a law, and it's about genes for round and a different version of the same gene that's for wrinkled, and it all works like this, and it seems very elegant but very reductionist. But now we happen to know that these wrinkled peas have more sugar and less starch in them than round peas, and that is because they lack a functional version of the starch branching enzyme that I mentioned. And so when the peas are forming and they're starting to stick together sugar molecules to make starch, they don't do it very well, and as a result there is more sugar left hanging around in the seed. And because there's more sugar left hanging around in the seed, they have a higher osmotic potential and they absorb more water as they're developing. And then when they get to the end of their life and the plant's senesced and they dry out, they lose that water. And because the water was a significant volume in that seed, they shrink and dry out and they wrinkle. So that is why the peas are wrinkled. They are wrinkled because they have a defective copy of the starch branching enzyme gene, and as we know, genes are transcribed into RNA message, and that RNA message carries the information out into the cell where it's turned into a protein. And so if you have a defective copy of the gene that doesn't make the RNA message, you don't make the protein. And so you slow up your starch synthesis because the protein would be normally involved in synthesizing the starch. And then you've got less starch and more sugar, and then the water accumulates, and then the seed drinks, and then it shrinks, and then it's wrinkled. So this is what's going on. And this is about the simplest kind of thing that could possibly happen in the context of a genetic system, because it's one enzyme right at the kind of end of a thing that is contributing in one very particular way. So this to me is kind of as reductionist as it gets. And even this to me is nothing about parts being sufficient on their own to explain the whole. It's about a process. It's about the interaction between a lot of parts and the consequences, the emergent consequences of that at a much higher scale, the scale of a seed versus the scale of the DNA. And so I think this genes for version, the reductionist version, is incredibly unhelpful because it doesn't really capture what's going on, and it's oversimplistic, and it underpins this kind of reductionist idea, which I think is entirely wrong in terms of how one thinks about genetics. And this holistic definition is much closer to what genetics is really about. It's about understanding that relationship between scales and how these higher order characteristics at higher scales emerge from the interaction and the intimate interconnection between the parts that underpin them. And so this notion of DNA as a blueprint, I think, is really a problem. It's a very popular conception of these, nucleotides and all the rest of it. A molecule of DNA by itself does absolutely nothing. And if you gave me the entire sequence, it would be, at this point, impossible to predict with any accuracy what would happen. And in order for that DNA to be read out, you need to put it into a cell. The cell has a huge amount of information in it in the form of all of those proteins and other components that are required then to transcribe and translate and all of those things. So there's a sort of chicken and egg issue here in that the DNA makes the cell and the cell makes the DNA, and you cannot separate them. And so I think this blueprint idea is very unhelpful. And then going even beyond that, the notion of a gene, it's a very useful concept that works incredibly well in genetics, but it tends to conjure up this idea of a kind of beads on a string of distinct chunks of DNA that are one gene and chunks of DNA that are the next gene. And you think about the genome and all of the genes collected as a whole load of these genes neatly arranged on a string in the genome. And this, as we've learned more and more about DNA, is incredibly far from the truth. So apart from Gregor Mendel, another one of my major heroes in genetics is Barbara McClintock, who did this extraordinary work in maize, determining through a combination of looking down microscopes at chromosomes, so I'm really observing the behavior of chromosomes, and correlating that again way up the scales to the appearance of kernels in maize cobs. And she, through that really careful work, discovered these things called transposable elements, or jumping genes. They are bits of DNA that move around the genome. They cut themselves out of where they are and they stick themselves in somewhere else. And if they stick themselves into another gene, which they do quite frequently, they can disrupt the function of that gene. So in this maize cob, the gene that makes the kernels this dark red colour is working in this kernel, but not in this kernel, because in this kernel there's a transposable element stuck in it. If it jumps out, then you'll get this early in the development of the seed, you'll get this entirely red seed. If it jumps out late, then you'll get these little patches of red on a yellow background. And the place where many gardeners have seen this is in Antirrhinum. Antirrhinum snapdragons, they have a particularly active transposable elements, and a lot of the horticultural varieties make use of that to make these kind of variegated flowers. So Barbara McClintock discovered these jumping genes, and we have since, through understanding more about genomes in plants in general, come to realise that a lot of genomes, particularly those in our crops, are absolutely stuffed full of these transposable elements. So this is a bar chart showing the genome of wheat. Now, already, it's important to point out, wheat actually has 3 genomes because it's a hybrid between 3 different grasses. And those genomes consist primarily of amplified up transposable elements. So all of these coloured here, versions here, are kind of different kinds of transposable elements. There's some just repetitive DNA in there that is probably not a transposable element, it's just very repetitive. There's some stuff that is labelled here as unannotated. And then this tiny yellow bit at the top, that's the bit that you would classically think of as genes. That's the part of the genome that is transcribed into RNA, and that RNA then makes protein. Plenty of the genome is transcribed into RNA that doesn't seem to do very much. I suppose what we've learned overall from all of this extraordinary work that's been enabled by the molecular biology revolution is that genomes are incredibly messy, ugly things. They are not beads on a string. They are incredibly complicated, strange things that have clearly accumulated all kinds of stuff that some people call junk, but some people don't like calling it junk because you don't really know what its function is at some level. But it's not straightforward, and it's certainly not the simple beads on a string idea. And just to kind of loop back to Mendel's round and wrinkled peas, it actually turns out that the mutation that causes wrinkle, that results in the defective gene for starch barging enzyme, is caused by the insertion of one of these transposable elements. So, and these transposable elements are only one source of variation in genomes. There are, over evolutionary time, many differences and changes that accumulate, genome rearrangements, errors during the replication of the DNA that get passed on, environmentally induced DNA damage, errors whilst the chromosomes are recombining at meiosis and swapping arms, which they do, errors in the repair of DNA that's been damaged, and then hybridization, like in the formation of wheat, whole genome duplication, and quite a lot of evidence for horizontal gene transfer. And indeed, transposable elements are arguably, and there's a good hypothesis that they are derived from viruses. And a lot of viruses introduce their DNA into the host, or their RNA into the host, and that is can, in some viruses, insert itself into the host genome. And so there are, through viruses, the possibility of carrying bits of DNA about the place between plants, for example, and many of those viruses are quite promiscuous from a species point of view. So there's evidence of gene transfer in that way. So it's very messy. It's definitely not beads on a string. And I'm always I like to contrast this with this idea of that nature is genius, and they're rigorously tested and find over millions of years designs. A lot of these processes happen over short time frames. So if you had cornflakes for breakfast, almost certainly every single one of the flakes was different from every single other one of the flakes because of the insertion of a transposon, because there are thousands and thousands of those in the maize genome, and although they move very infrequently because there are thousands and thousands of them, genomes are just not very stable, and yet they work. And it's a good thing that they're not, because it's this mutation, this change, that underpins evolution and diversity. The difference between Watson and Crick and a chimpanzee is an awful lot of mutations that have accumulated in those two lineages from their last common ancestor. And how those lineages are selected effectively, which ones survive, is the subject of Darwin's kind of assembly of the evidence that the way it can work is through the survival of the fittest, natural selection. So you've got all of this diversity being generated by all of those mutation processes, and any mutations that give you an advantage will survive, and those animals will survive, or plants will survive, and that will be passed on to the next generation. Any mutations that give you a disadvantage will reduce your chances of surviving, and any mutations that don't really make any difference will just have a random chance of being passed on or not, which is how you then wind up accumulating mutations that don't make any difference, and at the same time selecting out the ones that help you. Now, if you're a plant, one of your main problems in life is that you're rooted to the spot, and so you're incredibly vulnerable to predation. As every gardener knows, these things are vicious, and so many of the survival strategies of the plant are very strongly aimed at not being eaten in a whole variety of ways. So poisonous, spiny, tough, indigestible in a whole variety of ways, all of these kinds of things. And that's great, unless you happen to want to eat the plant, in which case it's really not a good idea. And so whilst survival of the fittest is great, what we extraordinarily did about 10,000 years ago was discover or come up with the idea that we could, instead of going with what nature had given us, go with what we would prefer. So survival of the favourite, and that's what domestication is all about in our crops, in all kinds of animals, both our farm animals and our companion animals, dogs, cats, all of the rest of it. And Darwin also did a huge amount of work on domestication. And so whilst natural selection is about survival of the fittest, it's about the plant, in this case, allocating its resources to maximize the number of viable offspring it makes, and for those offspring to have to be able to compete well with neighbouring plants and so on. And what that gives you are these kind of complex, dynamic, multi-scale regulatory systems that I've talked about, with all of those thousands of genes that they have, and they deliver flexible, adaptable growth habit, all kinds of defences, ability to disperse the seed off the plant, and also every seed minimally resourced. Whereas if you're trying to make a crop plant, those characteristics are actually a disaster. You'd like a much less flexible growth habit where you can harvest them easily. You'd like the seed to be delicious, nutritious, digestible, wonderful. You'd like it kept on the plant so that you can harvest it easily, and you'd like all kinds of good stuff to be put into each one of those seeds. And in return, so to speak, you're going to take this plant out of competition. You're going to give it a lovely, nice field with no weeds because you've got rid of them, and you're going to fertilize it and all this stuff. So very different pressures for natural selection and human selection. And you can see that very clearly in maize, which I showed you already. Maize was domesticated by extraordinarily ingenious Mexican farmers over thousands of years. Lots of evidence for masses of very deliberate crossing to wind up with what we see in modern maize. This is teosinte, which is its closest living wild relative. They're still actually cross-fertile. And you can see this is not nearly such a good idea from a crop point of view as this. And if you go and buy sweet corn in Sainsbury's, as I said, it's naturally sweet, which is a really good thing because it's natural, but it isn't naturally sweet. It's incredibly unnaturally sweet because in nature, in teosinte, the plant is not at all keen on its seed being eaten, and it's relatively tough and not nearly so easy to eat, and it's definitely not sweet. This is an unnaturally sweet sweet corn because plants really don't want you to eat their children. So in driving domestication in this way, we have over the years made use of this extraordinary variation as it arises to pick the versions of crop plants that we want, that are more nutritious and easier to grow and all of those kinds of things, and cross them all together to try and produce combinations that combine all the characteristics we like. And we've done this incredibly successfully. So here is this wild relative of teosinte, of maize, and here is modern maize. They look very different from one another because as part of the domestication, you've wound up with these single stems and big ears and of corn and all of this stuff. And here's the wild relative, teosinte. Now, supposing your elite crop variety that's got all of these wonderful combinations is susceptible to a new disease, but you found a teosinte variety that is resistant, and you would like to make this maize variety, this elite maize variety resistant, the way you would currently do that would be cross them together, and you get a hybrid which would have half its genes from the maize and half its genes from the teosinte, and it would be, you know, all those lovely maize genes that you spent thousands of years putting together would all then be mixed up with the teosinte genes. And in order to get back to this maize crop, what you do is you take the hybrid and you cross it to that maize parent again, and then you've got 25% from the maize and 75% from the maize and 25% from the teosinte. You cross that to the maize, and you keep going and you keep going for multiple generations. And every generation you select for the guys that are resistant to the disease, so they still have this disease-resistant gene, but you're gradually getting rid of more and more and more of these teosinte genes and replacing them with more and more and more of the maize genes. And you do that for multiple generations, and then you've done it. And that's how quite a lot of classical breeding works, and it takes a lot of time and a lot of generations. And so and on top of that, you're limited to the genetic variation you can find in crops that or plants that you can cross. And so to kind of increase the amount of variation available to breeders, for decades now, people have used deliberate mutagenesis to kind of add new mutations into the genome, and some of those will be useful, and then you can breed from those. And then on top of that, these new techniques, both genetic modification and now genome editing, people have got particularly excited about, because where you know what gene it is that you want to add, so supposing you actually knew what the gene was in the teosinte that was conferring that disease resistance, instead of going through those multiple generations of backcrossing, in principle, you could just take the gene that conferred the disease resistance and put it directly into your elite maize line, or change that gene that was in the a version of a gene that was in the elite maize line already, by editing it for a version that behaved more in the way that you wanted. So that's why these techniques are potentially so useful in situations, and only in situations, where you know what gene it is you want to change, and where it's only really one or a small number of genes that you need to change to get the effect you want. It can save you a lot of time. Okay, so what does all this mean? I think, I hope I'm convincing you that the notion that science is inherently reductionist and it's sort of purely rational and nothing to do with intuition is wrong. And in my view, particularly this thing I find frustrating, because in my view, the more you know you don't know. And the more you understand about how plants work, which is just extraordinary, certainly the more kind of humble I feel. It's amazing what they can do, and it's amazing to think about how that works, how those incredibly intricate systems with with the DNA and the RNA and the proteins and the cells and the tissues, and how that works is just extraordinary. And particularly when you think about photosynthesis and how plants are literally building themselves out of thin air, using that kind of machinery of the DNA and the RNA and the proteins and the cells and the tissues. So I don't really think of science as an activity that kind of crushes one's astonishment at nature and one's wonder at nature. It's very much a holistic activity, huge amounts of intuition involved, and I think it makes one incredibly humble. Whereas I think it is possible to be a person on this side of the line where your intuition is very dubious. You know, this natural food must be good for you because obviously plants are being put on this planet for us to eat. That doesn't seem to me to be a very humble way of thinking about it. And it's not, you know, plants are part of nature and they don't want to be eaten. So I think there's a certain twist to how you can think about things on that side of the line that's also problematic. And I would argue then that people on both sides of that line have to stand back and ask themselves, what do they really think about these systems? How can they really interact with them, understand them, engage with them in a much more positive and nuanced way, to think about almost some of those Prince Charles quotes. How do we work with the grain of nature? That's a very viable question for both those sets of people. And we're back to the point that that actually fundamentally all of these people are people, and they share a huge number of values. And so how are we in this position where GM crops are so controversial and genome edited crops are now coming into that sphere? And what's been the consequence of that for our opportunities to use these things to deliver our shared goals? So just very quickly on the policy front, the way that the anxiety about GM has manifested itself in law in the EU is that on top of the regular rules for variety registration for new crops, which require you to generate that the new variety is distinct, so it's different from other ones, that it behaves itself through the generations, it's uniform and stable, and that there is some advantage to growing it. Usually, I mean, it's a relatively small number of things that are typically assessed in this value for cultivation and use test that are to do with yield, for example. So that's what you have to do for any new variety to get that registered for commercial use. But then there's this great line in the rules that says there are additional rules for genetically modified plants. So you do that, and then there are additional rules, and there kicks in this huge range of safety tests and so on and so forth that you have to do. And nowhere in this are the things that people care about really assessed, as I'll get to shortly. So the EU law is very much based on this kind of social identity element, that natural thing. Any approach to a crop genetic improvement that alters genetic material in a way that does not occur naturally is counted as GM. And these random mutagenesis techniques I've mentioned have been exempted on the grounds that they've been around a while, although they're acknowledged that they could fall under this category. And at the moment, genome editing is still in hot debate in a lot of countries, and a lot of that hot debate is around whether these mutations could occur naturally. It's a really peculiar set of arguments, because we're talking about trying to answer questions about the safety of a crop based on how it is made. And if you decide that's GM, then you have to test it in this way. And if you decide it's not GM, you don't have to test it. And that seems to me to be fundamentally extraordinary, when what everybody wants is this reliable supply of safe food. Surely this is what we should be asking, not how do we make this crop, but does it deliver these characteristics that people care about? And a reason, I think the main reason we're in this complicated situation is because of these deeply embedded views about what it means to about the kind of trustworthiness of both big business and science, and these two elements that have embedded themselves in the law. So you can't trust big business to deliver on these goals that everybody shares, and also this idea that natural is good. Now, I hope I've convinced you that the idea that natural is good is actually perceiving of nature in a very human-centric way, which is unhelpful. And if we want to deliver these kinds of values, then we need to shift the way we think about our food, not natural is good, but rather those values matter. So I think these are the things that we really need to think about in the context of our regulation. Regulation should address these things. How can we provide a system that will ensure that the seed that gets sold commercially deliver on those values that I listed that everybody agrees matter? And I think we can do that by taking the current registration system, applying it to absolutely all crops, and taking out any idea that using a particular method is good or bad, and then layering on top of that the distinct, uniform, stable value for cultivation and use. Much more emphasis on that value for cultivation and use. Is that delivering on the things that people care about? And then include a tiered safety assessment based on risk from this value for cultivation and use idea, from what the crop is and what it's supposed to be doing, and what the safety and environmental consequences of that might be. I think that would be a much more robust approach that would genuinely support the goals that most people share in common, and it would give us access much more straightforwardly to the contributions from plant genetics to delivering on these common goals, always remembering that these common goals are actually only a tiny part of the overall food security question. And here again is a place where I think it's ironic that these kind of arguments about reductionism come into play. The entire argument about food security has been sucked into a debate about genetic modification, as though that were more than a very small part of the system. We all have to work very hard to deliver on those shared goals, particularly in the context of food security, and it's going to involve an awful lot more than plant genetics, but that's a huge problem, and we cannot throw away the benefits of plant genetics to delivering that. So I will stop there and take any questions that anyone might have. And I think if I click that button, things might happen. Now, oh, here we go. It's, I don't know. Have I stopped sharing my screen? And if I haven't, how can I do that? I can't hear you, Max.
Can you hear me now?
Yes, I can.
Yeah, I'm jumping in. I wasn't expecting to have to do this, but Julia seems to have vanished, so maybe she's lost connection. So what I can do is I can step in and take over. Julia, do reappear if you're there, but in the meantime I shall carry on. So we do have some questions. First of all, thanks very much for a fascinating talk, Ottoline. Really thoroughly enjoyed that. Let me have a look and see what questions we have. Well, actually one question that's quite interesting is where you ended up there, talking about values and goals and sort of having a much more sensible approach to all of this. What do you think the chances are that we might be able to sort of retrieve this situation? It feels like a situation that's, you know, gone in a very wrong direction, and what you were just saying seems very sensible, but, you know, what do you think the chances are that we can resolve this?
Well, you know, I'm an inherently optimistic person, and I think I do find it quite frustrating because the debates come up over and over again over many years. And there's always, every time, the opportunity to start from those common goals and to talk about those and to build on that. And somehow it's very difficult to square the debate, or to sort of focus the debate on those common goals and how best to achieve them. But I think that is fundamentally the solution. It's going on all over again at the moment in the context of genome editing, because there are a lot of people who are very keen to get genome editing taken out of the GM regulations in the UK, and that's fine, except for the fact that restates the debate in the old format. And in some ways reinforces the idea that the current laws are a good way to do things, and I'm finding that a bit frustrating too. So I think you're right, it's hard, but I also think that it's not impossible, and one has to keep trying.
Thank you very much. I'm going to bring Julia back in because I see Julia's online again, so just bear with me while I bring her up on screen.
Yes, sorry, my Wi-Fi completely went down at that point, so I sort of lost about the last 5 minutes, I'm afraid. So if I start repeating myself, please just tell me. Okay, now there's a, let me just, oh.
How are you doing, Max? You could throw me another one whilst Julia is
Sure, I will do. Disembodied voice here. Does GM still prevent farmers from using last year's seeds? It used to be said that was Monsanto's ploy to boost profits.
That's a very interesting question, in that an element, I mean, there are a number of ironies in the way that things have rolled out from a legal point of view. So because GM has been regulated in this different way as other, and it's been expensive for companies to get seed through that regulatory process, the rights that companies are able to hold on those seeds are more and different than the ones that they can hold on conventionally bred things, where there are these typical plant breeders' rights, so you're allowed to use them in, reuse them in breeding and all of those kinds of things. So the rules about what you can and can't do with GM seeds are different and give companies more rights than regular seed, because of the anxiety about them and the fact that they're separate, and that's not a requirement. And I think that can also be changed very straightforwardly in the law. I think I don't think there's any justification for that now. That's different from the other component that people quite often talk about, which are these terminator technologies, where the seed doesn't germinate. Which was supposed to be a way to reduce escape, so to speak. And there's lots and lots of press about that, but it never worked very well, so it never actually got used.
Here is a very quick one, just to keep things moving along while we wait for Julia. Which countries are most relaxed or strict about GM?
Well, that's an interesting question. There are different types of law in different places, and the law that is kind of closest to the sort that I think needs to be there, which is regulating not the way you made the crop, but what the crop does, is in Canada. But they still have, their law is still it has a kind of novelty component, so it only kind of gets triggered if there's something novel, and that novelty typically is defined in a way that includes GM and not other things, not entirely though. So a really good example is the problem that, I mean, actually, because the technique of GM was invented before we knew much about the genes, most of the applications that are out there in the field come down to about two different things, one of which is resistance to insects, and that comes from a gene from bacteria that the plant produces a protein that is normally produced by this bacterium, and that kills caterpillars that are eating the plant. So it's called Bacillus thuringiensis toxin. And so there are lots of crops with that in it. And then there are lots of crops that are resistant to herbicides, so that farmers can spray the crops with herbicide to get rid of weeds more effectively. And you can make those herbicide resistant crops either using GM approaches or using non-GM approaches. And if you make them using GM approaches, then you're instantly into these enormous safety tests and so on. And and from an environmental point of view, there are genuinely interesting questions about that I think need to be thought about. And so those Roundup Ready seed from Monsanto, they're in that category, and they're very widely grown in a lot of places, particularly for things like soybean, where weeds are a huge problem. But you can also make those with conventional approaches, and there you don't have all of those regulatory hoops to jump through, and you don't have to do the environmental safety testing and so on. So I think that's a good example where this current approach of regulating based on the technology is particularly strange. And then which countries? So most of America, both North America and South America, grow substantial proportions of those two types of crop, particularly soybean, as I say, because it's so such benefits from the weed control elements. So it's actually really quite difficult to get non-GM soybean internationally, and soybeans used hugely in animal feed. So that there's been lots of debate about whether it's acceptable for organic farmers to feed their cattle GM soybean because they're having a hard time sourcing non-GM soybean. So countries otherwise, very few in Europe. There's one licensed crop that's grown in Spain, relatively little across Asia still, but yeah, so America, I guess, is all of the Americas the most common.
Thank you very much for that. I'll just do one more and then see if I can hand back to Julia. A question relates to the fact that you were explaining how complex the genome is and how in many ways our understanding is fairly rudimentary, and we have now this relatively new technology of genome editing. I was just curious on whether or not we get unintended consequences from using this new technology. Is that becoming a pattern or not?
So unintended consequences are really a very interesting thing, in that all of those approaches that I talked about, conventional breeding, GM, genome editing, all of those things can result in things that you weren't expecting, let's put it that way. I don't know whether that's unintended consequences. They're also called so-called off-target effects. So you thought you were carefully back-crossing to make sure you only got the bit of the genome that you were interested in a regular breeding, but you've also brought with it the surrounding DNA and actually probably some other DNA that you weren't quite sure what it was from elsewhere. So you've definitely, you've done things you don't know. With GM, where the gene lands is of interest. You don't really know what that might or might not do, and there's some evidence also that when you're introducing the DNA, you can wind up with it was kind of thinking about inserting into the genome somewhere else and then didn't. So you can pick up extra genetic changes that way too. With genome editing, there is some evidence for off-target effects, so events elsewhere, and for bigger changes where you were targeting the change than you were expecting, that take out flanking DNA, for example. So absolutely all of these things are imprecise in various ways. The degree of precision, I would argue, increases up that spectrum. You've got less probability of having done something unexpected with genome editing than with GM, than with conventional. But there's an interesting debate in the field because we've been doing this for thousands of years without having a clue what we've been doing, whether now is a good time to start asking all new varieties to be for those kinds of things to be checked out. Should we sequence the whole genome of all new varieties? I personally think that's overkill, not least because of all those transposons, which means that anyhow that the idea that there's some kind of stable genome that you've captured by sequencing it once is not very accurate. So nothing. Yes, I guess that sort of covers it. There could be off-target effects, but no more than normal.
Thank you, Julia. Can I maybe try and bring you back in again?
I think I'm here.
Yes.
Sorry, the router's been flashing horrible colours at me. I don't know why it's doing it on such a nice evening. It's usually when it's snowing or something. Now, I'm not entirely sure which questions you've already answered. That's the problem here. Somebody just recently has made the point that the problem with working from a risk perspective is that people just don't understand risk, and they're drawing parallels with all these vaccine debates and AstraZeneca and so on. You know, the risk is minuscule, but politicians leap into action and ban things. So there are strong parallels there with the sort of attitude to GM as well, aren't there? But how do we move away from this apart from trying to change the perspective that the public have of scientists? I mean, what could we do in the next couple of years to improve the image of science in relation to crop breeding, for instance?
So I think, I mean, the way I see it is it comes from all directions. I think scientists need to think hard about their image too. I don't think that deficit model is helpful, that people don't understand things and so we need to explain it to them very slowly. I think I'm more interested in these open conversations that start with shared values, and because there's a lot of evidence that things like ability to assess risk and so on, what really matters to people is trust in the governance arrangements. And where those governance arrangements are built on the shared values, then I think you have a much better possibility of getting to that kind of situation. So I think what's needed more is to build trust relationships based on people right across the system, scientists, communicators, members of the public, talking to each other, rather than this notion that we have to make people understand things or educate everybody. So it's not that education. I mean, I love education, but what you need to do is give people that sense of personal agency, not do stuff to them, tell them what they should think. And I think that the longer we say we must convince people that, the less likely we are to do it, because that's not a good place to start a conversation.
Yes, very good points there. Now we have a really long question here. I'm not sure who it's from. They say that they think that GM techniques are incredibly useful and powerful tools, but current application of GM crops and investment into research seems to be quite short-sighted. And the author of this question thinks that's where a lot of the opposition comes from. While it might be possible to hugely boost productivity with a Roundup resistant crop, for instance, soil erosion and loss of biodiversity don't seem sustainable for guaranteeing food production in the long term. And of course, pests quickly evolve resistance to resistant crops grown in monocultures. So this questioner says it seems to them that agroecology and growing diverse crops is important for food system resilience and sustainable production. There might be less opposition to GM if GM crops were used in more agroecological systems than in profit-focused mega monoculture models, where local varieties, knowledge and genetic diversity are often lost. So that's a very long question.
Yeah. Well, at some level, I think that that's sort of what I was saying, in that what people care about are that I have a slide of these kind of shared values of the kind of agriculture that, as far as in my experience, is what everybody wants, and that is partly what's being described by the questioner, an approach to delivering that kind of sustainable agricultural system that is providing a secure supply of nutritious food in a way that is environmentally sustainable, and where the risks and benefits are shared equitably. And a lot of the points the questioner was anxious about with you know, big business and particular applications and so on and so forth are you know, negative points against that list. And I think if we have a regulatory system where that element in the middle, that's currently called value for cultivation and use, asks those kinds of questions, asks about environmental sustainability, asks about the protection of soil and pesticide reduction, and if those are things that are underpinning the regulatory system, those questions are actually asked in the process of regulating new seed varieties, then I think that's how we can move forward to that shared vision for agriculture that people want. And at the moment, that value for cultivation and use box is really quite a limited set of things that are included. And I'm very interested in how we expand those things and incentivize breeders, apart from anything, to think about that wider range of characteristics, and to think about breeding for not for monoculture but for mixed seed populations, all of that kind of stuff. There's a huge amount of rather exciting innovation that could happen if we shifted both the regulatory system and the kind of incentive systems in the whole supply chain.
Yes, I was just thinking actually about mixed cropping. For instance, in Kenya, for instance, maize and beans are often intercropped together, and therefore you wonder how crops, for instance, maize, which is tested on its own in beautifully well-controlled field sites, how that performs when it is actually grown in a mixed cropping system.
Yeah, I mean,
with competition and all those things that go on in that setup.
Yes, I mean, I think part of the issue at the moment is that the breeding systems are set up in the West to support monoculture, and that's how they're tested, and they're tested in fixed environmental conditions with particular amounts of fertilizer and so on. And so again, the incentives to deliver a wider range of characteristics in the crops are not there. I think there's always, I mean, I think we need an evidence-based approach to what the most sustainable agricultural systems are, from the point of view of maintaining fertility in agricultural land and also protecting biodiversity. There's a lot of debate about whether a better idea is to farm relatively intensively on a small area, or freeing up land for biodiversity, or whether it's better to farm unintensively on a larger area so that your biodiversity is coexisting with the farming practice. And that's quite place specific because it depends on all kinds of characteristics. So, you know, there are things we all want, which I've listed on the slide. How you deliver those is different in different places and with different circumstances and different demands from the population on you know, on the time people are willing to invest in harvesting, for example, and the amount of money they're willing to pay for their food. All of those things, all the behavioural science and so on, we have to factor in to try to work towards these systems that are what, as I say, people typically want. And the regulatory system and the dialogue around food and farming need to match those ambitions, not get dragged into an endless sink debate about one tiny part of it. It's quite frustrating.
Yes, all very good points. Now, I'm just going to ask a question which Max typed in a long time ago this evening, which is that, is the distrust of GM becoming less of a problem for most people? And he wonders if there's a highly vocal minority that gives a false impression of public resistance to GM.
So again, typically and there's a lot of really high quality public dialogue and engagement activities that have happened around GM. And it is typically the case that when you have those longer conversations, it emerges that people are comparatively unconcerned about the technology itself. There are always some exceptions. There are some people who simply are not happy with the idea. That's their opinion and it's fine. But typically people are more concerned about what you do with it than the technique itself, which is one of the reasons I'm so convinced that if we can shift the regulatory system to support ideas about what we want to do with our farming, that would go a long way towards allaying people's fears. But because we're endlessly talking about the technology rather than what we do with it, you tend to wind up just reinforcing the old debates rather than moving forward.
Yeah, very good points. Now, just one last and extremely short question, which is from one of our anonymous participants, and they ask, does Mendel's wrinkly gene segregate in natural plant populations?
I don't know. I'd be amazed if somewhere in the world there wasn't some species where that gene hasn't randomly mutated, resulting in that segregation. I don't know enough about, I should do really, historically, where Mendel got all his peas from in the first place. But
no, well, perhaps we ought to leave it there for this evening. And thank you, Ottoline. This has been a stimulating and very thought-provoking presentation this evening. I'm going to be going away and sort of mulling over a lot of the different things that you've brought to our attention tonight. So thank you very much. Normally, and if you'd come to Edinburgh, we'd have taken you out for a Chinese meal now, and I'm afraid that can't happen. But anyway, and I'd like to thank the audience who've come tonight and who've asked so many interesting questions. So thank you, everybody, and thank you, Ottoline.
Thank you.